Multiple myeloma is a cancer that affects 30,000 people every year. Cells of this cancer have genomic alterations from the typical human genome. These are called variants. When working with 70 cell lines of this cancer, we found it difficult to find a reference database cataloging variants that weren't small. Hence, for 3 other types of variants, we first ran bioinformatic tools to identify these variants from sequence data. Then we created a database to characterize and visualize these results. Cell line reference databases are often static table, but our databases are queryable, shiny, or web app.
This is our Shiny dashboard. A researcher can use our dashboard to get information about cell lines. For example, if they select cell line INA6, the distribution of variants along the reference genome in this particular cell line is displayed. This can help researchers in identifying mutation hotspots before carrying out biological experiments. We also provide overview visualizations of all cell lines. For example, a researcher can see the number of variants and the type of variants found in each cell line. Using this plot, a researcher can obtain a list of top mutated genes across all cell lines. Researchers can get similar information and visualizations about other types of variants, such as structure variants and copy number variants, in these cell lines using other tabs. Another important feature of this dashboard is to help researchers select genes to knock out in their experiments. Let's say the researchers choose to knock out SFPQ and KRAS gene. Our dashboard will dynamically calculate the mutation burden, which indicates the total number of mutations found in that cell line. Our dashboard also provides the burden change after knocking out the selected genes in this cell line. By sorting on this metric, researchers can select the cell line that is most affected by knocking out these genes.
By characterizing these cell lines with 3 variant types, our database will enable researchers to identify new candidate drivers genes and therapeutic targets. Our database's queryable dynamic interface will catalyze cancer research by reducing the time and coding expertise needed for researchers to find information about these cell lines.